All right. Thanks very much to the organizers for putting on this great workshop. It's a real privilege to have the opportunity to present here, and thanks to everyone for staying until the last talk. Yeah, there won't be anything too complicated in this talk. So this is based on joint work with my PhD student Hae-Bin Sung. So this is pretty much her work, and so I'll be talking about variable selection and classification with presence-only data. So what I mean by that is that normally when you have a two-class classification problem, you have positive and negative labels. But in a number of applications, and so this is something that I only discovered a couple of years ago, there's many scenarios where you kind of have positive samples and then unlabeled samples. So these are samples that you don't really know whether they're positive or negative. And so in the machine learning community, this is referred to sometimes as one-class classification or PU learning for positive and unlabeled data. And so these arise in a number of applications. So you'll see a couple soon. So population ecology, information retrieval medicine. I'll talk in more detail about sort of the application that motivated this work in biochemistry. And so the question that we're interested in here is can we classify and select variables using sort of the presence-only data? And we'll see later on that a number of sort of algorithmic and statistical challenges arise because of the fact that we have these unlabeled samples. We'll see that they're sort of not too bad and go into sort of how we address that. So just to present a couple of applications to motivate it. So this is not my application. This actually comes from work that sort of Trevor Hasty did at Stanford with one of his PhD students, Ward, about 10 years ago. So he had a collaborator in Australia. And so this guy sort of works in population ecology. And so one sort of question that people in Australia who work in sort of population ecology is sort of tracking species that are, that they're interested in. So in particular, one sort of species that a lot of people sort of get excited to see in Australia are koalas. So, people are interested in sort of where you can find koalas. So, if you're interested in seeing them, this is sort of a map, I guess, currently of where, locations where they're typically observed. And so this is sort of a very natural case of presence-only sort of data, because you can imagine that, if someone has seen a koala in a particular location, then you can certainly mark that as a positive. But if, a koala has not been spotted in a particular location, then I mean, you don't know that there has never been a koala there. It's just that it hasn't been spotted. So this is kind of a clear case where you have sort of positive and unlabeled data. So this is sort of a good, simple example to sort of keep in mind. So the application that actually sort of motivated this work sort of came from a collaborator in biochemistry. So a very fundamental question in biochemistry is how to relate sort of protein sequence to function. So proteins, if you think about them at the sequence level, can be thought of as kind of sequences of amino acids, and sort of all proteins sort of have some function that they're sort of meant to perform. So whether they're meant to increase the rate of reaction, or whether they're meant to whether they're meant to catalyze a particular solution. So there's many different cases where you want to sort of look at the function of a protein and sort of determine how that's related to its sequence. So there's really two sort of questions that people are interested in. So from a scientific point of view, you're interested in sort of understanding how does the sequence relate to the function. And then from an engineering point of view, you're interested in, can we actually use the knowledge that we have about the sequence to actually create a better sequence that kind of improves the functionality of that particular protein. And I'll go into a specific example later on in the talk. So presence-only or positive and unlabeled data come out of this particular application, because in some sense this is to do with the sort of the limits of the technology in sort of high throughput sequencing in biochemistry. So because we're interested in sort of function, that sort of typically means that a chemical reaction has kind of happened or not. So it's sort of fairly easy to detect that, or more straightforward to detect that kind of a chemical reaction has happened, and so sort of positive or functional sequences can kind of be collected pretty easily. But it's quite hard, or sometimes impossible, to actually collect negative or non-functional sequences. And so there's sort of more to this experiment, which I won't really go into. But essentially, you end up with, you know, a whole bunch of, like, you know, on the order of thousands or millions of unlabeled sequences, which you don't know whether they're functional or not, and then, you know, positive sequences. So these you sort of know to be functional. Okay, so this ends up being a you know, a large-scale variable selection problem when you have this sort of positive and unlabeled data, because you could imagine that, you know, a protein has sort of D positions with you know, where each position can be one of roughly 20 amino acids. You know, there's a number of different models you could imagine fitting to it. So if you fitted a main effects model, you'd sort of have order of DM variables. In our particular example, D would be on the order of maybe hundreds. If you wanted to fit pairwise models as well, you'd have D squared, M squared, and then if you want it to be as general as possible, you'd have M to the D. But what you can kind of see from this is that, you know, the variables kind of blow up pretty quickly in this case. And so the observed data here are the X's, which are the sequences that you observe, and they're corresponding, I guess, sort of pseudo-labels, these Z's, which are 0 or 1, where 1 kind of indicates that it's a positive and you know that it's a positive, and then a 0 implies that it's unlabeled and you don't really know whether it's positive or negative. And so some important sort of parameters or quantities that kind of come up here. So these aren't really parameters, but quantities that sort of play a role are the number of labeled sequences, the number of unlabeled sequences, and then you need to have some information about, you know, the unlabeled samples. And so what we assume here, and what turns out to be true in this particular setting, is that we know the probability of you observing a labeled sequence, so the probability that y equals 1. So this is something that we actually know to within, you know, 5, 6 decimal places from an alternative experiment. There's work that, you know, Trevor Hasty and Will Fithian have done on, you know, sort of what you need, like, you know, whether on some sort of less restrictive assumptions you can make on the knowledge of pi. But for this context, we kind of assume that we know pi. And so we want to determine an algorithm for selecting which positions and amino acids influence the functionality. So this is clearly just a, you know, a variable selection problem.
Great. So pi is just a number, it's not depending on x?
No, it's just a number, yeah. Yep. Yep.
The sequences, they come with different lengths. So here you assume that they are aligned.
Yeah, yep. So we're assuming that they're...
cut them kind of like on the...
Yeah, so we cut them.
The same size.
Yeah, they're all the same size, yeah. So we're assuming that, yeah, they're aligned in the same size, yeah. Okay, so a pretty standard statistical model for classification is logistic regression, because this allows us to do both variable selection and classification, which is what we're interested in doing. And so the data we see are these xi's and zi's. The yi's, which you could think of as the true labels, they're unobserved. And so we have that, you know, if zi is 1, then we know that y is 1. But if zi is 0, then we don't know what the y's are, and the probability of that is pi. And there's also, like, something that you know, how these unlabeled samples are generated is fairly important. And I sort of won't go into the details of how, you know, all the math comes out. But what we kind of assume is that the zi's are actually the two populations are kind of drawn from the original population, but at different rates. So the unlabeled samples, which correspond to zi equals 0, kind of are drawn from the original population, and kind of the zi equals 1 samples are kind of drawn from, you know, you assume that you sort of draw y's and then only keep the positive samples. And this is actually consistent with what occurs in our particular application. And so this is referred to as the case control setting. And so that certainly, like, this assumption, you know, influences the likelihood and the objective that we eventually deal with. Okay, so sort of moving on to how we solve this problem. So, you know, the most standard thing to do is to sort of write down the likelihood and sort of see what we get out of it. And so this was work that was done by Ward and Hasty and others when they were kind of working on this sort of, you know, animal ecology sort of problem. So you can write down the likelihood, and it looks like this. So So this is just looking at the likelihood given that you've observed the z's and the x's, and so you can see that you get something that kind of looks like a logistic regression, but it's a little bit different. And one thing to sort of observe and note is that the observed log likelihood is non-convex, and so this is kind of the price that you pay for having unobserved or hidden variables. So the y's are hidden, so we're kind of integrating over them, and so sort of going through the math, you end up with this likelihood, and then so. In some sense, in the original linear logistic regression, you'd get that f theta was just this linear function and z's were y, and then you'd sort of just get the standard objective. But we get something here that's a composition of a convex and a concave function. So this is not necessarily convex, and so.
You mean addition of a convex and a concave function?
Sorry.
You mean addition of a convex and a concave function?
It's the composition, right? Because it's like s is f theta is.
Okay.
Right? No? Because like we're kind of saying a of s is that original thing. It's like a of like f theta of x, right? It's like.
Oh, I see.
Does that make sense?
Yes.
Yeah. Okay, so this is the objective that we end up with, and so this non-convexity presents potential challenges that we'll deal with. Okay, so in addition to having this non-convex objective, we also need to select variables to incorporate some kind of sparsity through regularization, and so we do that through we could do that through L1, the simple L1 regularizer. We also looked at a group regularizer. So this is very natural for this particular application because you can imagine that for each position, there's a bunch of different variables corresponding to different amino acids, and you can imagine grouping them together to say that either that whole position is active or it's not. So we kind of consider both of these regularized, and so we end up with this regularized regularized optimization problem that where the objective is non-convex. Okay, and so this is a problem, as I mentioned, that had been studied, and so when we see hidden variables from a statistical point of view, the most sort of natural thing we think about doing was to just use the EM algorithm, and so if we can write down the full log likelihood in terms of the y's, which are unobserved, and this is obviously convex, and then just treat the y's as hidden, and then just iterate through this. And so the EM algorithm just looks like this, where at the E step we estimate the y, and then at the M step we just solve this regularized likelihood problem. Okay. And so this certainly worked well for Hasty and others when they were looking at this application of population ecology, because they had nowhere near as many variables and samples. But in our example, we have on the order of millions of samples and on the order of tens to hundreds of thousands of variables. And so it turns out that this algorithm didn't really work or didn't kind of scale to this size of data. So, and the reason for this was basically because the M step of the algorithm is too slow. So we made this pretty simple observation that we only needed a majorizer for the M step to ensure that the objective decreases at each iteration. So rather than using the original sort of M step, which involved this regularized likelihood problem exactly, we just use a quadratic majorizer for the M step, and then we also sort of got an additional speed up by kind of using by exploiting sparsity of the feature matrix, because the x's are sparse because they typically represent whether a particular amino acid occurs in a position. So we also exploit the sparsity of x. Okay, and so what we end up with is just another sort of a modified, I guess, EM algorithm where in the M step we just use quadratic majorization. So rather than using the original log likelihood, we just use the quadratic loss. And so the basic idea is that solving a quadratic, so solving a regularized quadratic problem is much easier than solving the original logistic, like regularized logistic likelihood. because we tried many different packages for the logistic problem, but we found that they were too slow. But because quadratic programs are much easier to solve, we were able to sort of solve that using pretty simple coordinate descent or blockwise coordinate descent methods. And so this is essentially just exploiting a slightly broader class of algorithms than EM. So EM is sort of part of this class of algorithms, which are just MM algorithms. So these are just majorization, minimization algorithms, where essentially, one of the key properties of the EM algorithm is that it descends at every step using the convexity of the likelihood, which follows from Jensen's inequality. So here we just use we essentially just use a majorizer that's not as sort of aggressive as the likelihood, and that sort of gives us this. It still gives us this descending property, but as we'll see, it's sort of much faster. Okay, and so what we can see is that we get a very substantial runtime improvement, so on the order of more than sort of 100 times faster, and this is sort of what we need to actually solve the original problem that we looked at. And then the additional sort of gain that we got computationally was that we could exploit the fact that x was sparse. So for the blockwise coordinate descent step for the M step, we used a QR decomposition, which we know that if x is sparse, that doesn't necessarily imply that the QR decomposition is sparse. And so we sort of modified that, and this gave us about another sort of 30 to 40% increase in speed. So I mean, this this is all well and good in that it allowed us to sort of develop a faster algorithm, but it sort of doesn't necessarily say anything about the quality of the solutions that were obtained. And so this is to analyze that, we kind of took a statistical approach of trying to determine how good each of the solutions is. And so a number of you can probably sort of see where this is going. That what we can do is this is potentially challenging because the likelihood is non-convex, and so there may be local minima. But as Andreas presented a very nice talk yesterday, and we had we've sort of seen that now sort of non-convex objectives aren't sort of too troublesome. So in particular, there's now sort of existing theory for analyzing local minima of regularized non-convex M estimators. So Poling and Martin did some work a while back, and Poling did some follow-up work, and there's work by May, and Montanari, and then as I mentioned, so Andreas and Sarah did some recent work, which they informed me is going to be published soon. So we can kind of use this theory to sort of see if if our objective kind of has nice statistical properties. And so one of the main challenges to sort of showing that we get nice statistical performance, and this was again something that Andreas talked about yesterday, is we need to have this notion of restricted strong convexity. And so if we can prove restricted strong convexity, which is kind of just saying that we're strongly convex in a kind of a local neighborhood of the true parameter, then we're going to be able to kind of recover the parameter, and it'll have nice statistical performance. So so we can sort of make assumptions that are pretty natural. So in particular, if we just assume the x's are sub-Gaussian with some parameter, and that the XX transpose matrix is positive definite. In Quentin's talk a couple of days ago, you mentioned that one of the key things you need to assume for when you're doing logistic regression is that the probabilities can't be too close to either 0 or 1. And the reason for that is you kind of end up in areas of low curvature, which hurt you. And so that's what sort of these sort of the next two assumptions kind of correspond to, just making sure that we're not in regions of low curvature. And then finally, you might imagine that we need some conditions on how much unlabeled and how much positive data we observe, and that needs to be sort of upper and lower bounded. So clearly we need enough positive, enough sort of labeled positive samples to get some positive signal. We also need enough unlabeled samples because obviously all of the labeled samples are positive, so we need to get some signal out of the the unlabeled samples, which tell us about the negative samples. Okay, and the statistical guarantee is kind of just it's basically sort of what you'd expect. We kind of get this standard variable selection performance, and we get a similar sort of result for the L1 norm as well. And this holds for kind of any stationary point of our algorithm. We show that our algorithm kind of converges to a stationary point. Okay, so the proof is it follows pretty standard techniques, and in Andreas' talk he sort of talked about some of the main challenges that arise. So in particular, we first used standard convergence guarantees for EM or MM algorithms to prove that we converge to a stationary point. And I mean, one key question is like how do you initialize or start? And it sort of turns out that you basically can start in anywhere that's sort of not terrible. And so what I mean by that is that when we tried random starting points, we generally got to a final solution that wasn't wasn't too bad. And so we're wondering if kind of this objective was sort of too nice in some sense. But basically, if we tried to be adversarial and choose a point that was actually bad, we could certainly come up with bad points, because you can get stuck in regions of sort of low or negative curvature. And so the main challenge in sort of, as I mentioned, in proving this result is to prove restricted strong convexity. And so the way that we do that is to decompose the likelihood into kind of a sum of a strongly convex term and then a non-convex term. And this is a pretty standard sort of approach in proving restricted strong convexity in these cases, that you can in some sense decompose it into something strongly convex and then add it to something non-convex and sort of show, and then the work sort of goes into showing that the strongly convex term kind of dominates the non-convex term, which occurs here, and we can kind of show that under the assumptions specified that this occurs like using sort of various techniques in sort of concentration bounds for empirical processes. So this is kind of the flavor, and these are sort of pretty standard techniques that are used to prove these restricted strong convexity for these non-convex objectives. Okay, and so this analysis also suggests that, I mean, the reason we kind of used this approach was just that the reason we used this approach was because we kind of just wanted to get an algorithm that sort of worked quickly for our particular application. But the analysis sort of suggests that there's theory to suggest that if we can prove restricted strong convexity along with some other assumptions, that there are many first-order methods like proximal gradient, projected gradient, other approaches that could potentially be used, and we can potentially prove algorithmic convergence rates as well. So this is something that I suspect we can do, but we haven't done that yet. But so this sort of suggests that it's really the niceness of the objective that kind of allows us to do that. And so this sort of really, to put it into sort of the context of a couple of other things we've heard about, this is really just saying that the problem of positive and unlabeled data is sort of, it's non-convex, but it's sort of not non-convex in a terrible way. It's like the problem of dealing with missing data or errors in variables in linear regression. You can kind of show that it's not too non-convex in a bad way, because it's sort of it's just a small perturbation away from something that is convex. Okay, and so just to validate that, our original goal was to develop a method that sort of worked in this particular biochemistry example. So to just take a particular protein of interest. So one protein that's sort of important and relevant in biochemistry is beta-glucosidase, or BGL. And so one of the reasons, so one of the important processes in biotechnology is decomposing plant matter into fuel, so to create sort of energy sources from plant matter. And so there's a number of steps in this process, but one of the steps is that we need to decompose sort of disaccharides into sugar. And so beta-glucosidase is known as a deconstruction enzyme, so it helps to decompose like disaccharides into glucose or sugar. And so the question is, can we engineer these BGL enzymes to improve ability to deconstruct into glucose? Okay, so the statistical model that we have here, so this particular protein has 502 positions, and as I mentioned, so 20 amino acids. So the number of labeled samples is on the order of 1 million, and the number of unlabeled samples is also on the order of 1 million. And then, as I mentioned, we can sort of estimate pi from from another experiment to be sort of 0.35. And this is data generated by my collaborator, Philip Romero. Who's at UW-Madison in biochemistry. And so we considered both main effects and a pairwise interaction logistic regression model. And so so we have kind of on the order of 10,000 variables if we look at the main effects model, and then and obviously sort of that number squared for pairwise interaction models. And so I guess like sort of in general, validating variable selections challenging in practice. So we used a few different validation schemes. One was just to show sort of classification, to show that it does okay on classification. Validating classification is hard when you have unlabeled samples, and so one very naive approach to doing that is to just treat the unlabeled samples as negative and see that you're at least performing better than random. And so you can't get an RAC value of 1 or get a perfect RAC curve in this case. And so we sort of see that our method does better than random. The other and the gray curve here is kind of what you'd get if you looked at the ideal RAC curve. Another validation that we did that's sort of important if you're doing variable selection is to just validate that your stability, that your variables are pretty stable that you select. So what often kills variable selection practice is that it's not very stable due to dependencies across the variables, which has been referred to in a couple of other talks. And so what we did is we just sort of measured stability through this naive measure of this Jacquard index that's between 0 and 1, where 1 indicates that you kind of have, that the two subsets are sort of the same, and 0 indicates that they're disjoint. And so the higher the value, the better. And when we averaged across several different cuts of the data, we saw that the variables selected were reasonably stable. And this sort of makes sense in a way because of the way the x's were generated pretty randomly, so the variables are fairly independent. Okay, and so I mean, in practice, the ideal sort of validation you can do is scientific validation. And so the way that this was done was to actually sort of use some variable selected to engineer a protein to see if you can actually improve the performance. So what was done is for a particular BGL enzyme, so a wild type or natural BGL enzyme that was sort of known to be functional, we used our approach to mutate or to change 10 of the positions in the sequence. And so this was done by my collaborator. And then what we can see is that if we compare the performance of the wild type sequence to the design sequence, that we kind of we sort of decompose sugar sort of a lot more quickly, kind of using these perturbed, this sort of mutated sequence. So here the normalized concentration of 1 is kind of ideal. And so we could use the variables that we selected from our method to sort of improve this sort of rate of decomposition. And so this is probably sort of this sort of suggested that our approach worked here. Okay, and so finally, in terms of one other check that we did just to compare to other methods was that we do reasonably well on classification if we sort of compare it to other methods on simulated data. So there are a number of other algorithms that are out there. So here are just here are sort of a few that sort of we found to be sort of state-of-the-art algorithms for doing classification with presence-only data. So in particular, there's work by Elkan and Noto on the bias-corrected logistic regression. And then they have a second algorithm, and then you could sort of treat like this a support vector machines a pretty natural approach to do classification. And because you end up with unlabeled samples, you sort of have a bias that is corrected, and so Liu and others worked on that. And then there's also work that sort of looks at an asymmetric loss function. And so what we see is that if we look at the performance of our algorithm, which is this PU-LESO algorithm, that it sort of compares favorably to these other algorithms. Firstly, in the low-dimensional setting, and so here the oracle just is the setting where you kind of have all of the labels. So clearly, that's going to do the best. So we want to sort of get as close to that as we can. And so we considered a number of different simulation settings here, like depending on sort of this sort of plots the weak, medium, and strongly separated cases, and there's sort of more details on that in the paper. In the high-dimensional setting, we can see that we still do pretty well, or do better than most of the other state-of-the-art methods. And this is it sort of has to be said that those simulation results were in a setting that where we used the data that was generated from our original model. So if we chose data to be generated from a misspecified case, so in particular we chose data to be generated in a way that sort of suited these other algorithms. So this is just one instance of that. There are others. We see that we kind of we didn't do as well as the other methods in all cases, but we didn't do too badly, and so we never do too badly in terms of classification performance. Okay, so that's pretty much all I have to say. So we developed an algorithm for variable selection using sort of presence-only data or positive and unlabeled samples. We developed this algorithm which used quadratic majorization and provided statistical guarantees for that, and then validated that in a real application and then on simulated data. And then there's some open issues. As I mentioned, I think first-order methods probably would work better here. And then from an application point of view, and what will present additional issues is that in many other cases we'll have missing values and also uncertainty in the variables, which removes sparsity, and this creates other kind of computational challenges which we're currently dealing with. Thanks very much.
So there are these methods with class conditional noise or random classification noise where you can essentially think of it as a corruption mechanism where for each particular class I can corrupt it with some probability. So here I guess you don't corrupt the positive labels, but you think of it as being given corrupted labels for the negative ones.
Right.
With these you have consistent estimation procedures. So maybe it's one of those other methods where so you can just essentially use a weighted SVM, and that would be consistent. You can get those weights, which it seems you do have weights. So have you compared those?
So I guess the weighted SVM, so this is like I guess these methods here. I guess the weighted SVM or bias SVM. So we did compare those for classification, and it seems to do better than that. Yeah. Is that what you had in mind, or?
I don't know. But there is a weighted SVM that's consistent.
Right.
But so that may also suggest
it's consistent in variable selection consistency or classification.
Classification error, but I think you can even show for parameters.
Okay.
I think, especially if you have a model like a logistic regression model.
Right. Okay.
But that might also suggest how to get to a good initial point.
Right.
But if you have guarantees for consistency, but they don't actually perform that well for various reasons, then you can use those to get to an initial.
Right. That's true. I guess we found that our initial point was it didn't matter too much. We started sort of with an intercept-only model, and the initial point didn't seem to change things too much. But that's something that you should look into. Yeah. Thanks.
So Garvesh, there's this in your koala example. You said, of course, if there's a koala in a place, there's some probability that you just won't see it. So that probability there, how does that affect your performance? Like, can you say something like, oh, well, if I have unlabeled samples or positive only samples, then it's somehow like working with n over 2 labeled samples? Like, is there a sense of how much you pay for having presence only data?
Yeah, I guess it's sort of captured in these quantities pi, like I guess, so it's sort of captured so yeah, I mean in the likelihood we get this ratio like NL pi over pi nu, and I guess to me that's sort of the closest thing in terms of how much you pay. So I guess like I think this should get close to, I think, like the optimal case I think is roughly like, you know, maybe 1 minus pi or something, but it's essentially captured in these quantities. Does that help? Or?
Yeah.
So my question is regarding the application to the amino acid sequences. So I mean, at the end of your procedure, basically you obtain this theta vector, isn't it?
Yep.
And then basically, how do biologists interpret those thetas? I mean, what do they?
I mean, I guess
What's the
like
meaning of a small theta or a large theta?
I mean, I guess like it wasn't. I mean, so we developed a statistical test to say which is the most significant. So the actual theta values were not used, but they effectively were used to say these are the most significant. And so that we haven't really come up with an interpretation of the values yet. We've just said that let's take the top 10 features and then use that to actually change the sequence. Does that make sense? Or so we haven't we don't quite have an interpretation for that yet, other than I guess the higher the theta value,
So, I mean,
or
this model, the amino acid, basically the amino acid enters kind of linearly, isn't it?
sorry?
In this model, amino acid enters linearly.
Right, yeah.
So that basically, I mean, but of course, amino acids, they kind of could be strongly correlated between the positions, right? For example, there are like, I don't know, conserved regions and so on.
Right.
Yeah. So
Right,
this
yeah, that's
model
true.
doesn't take into account those correlations.
No, it doesn't. Not in terms of the, yeah.
My second question is basically, if you change, if instead the way the amino acid, this variable enters in your model, if you change it to the function of amino acid, is that going to significantly change anything here?
The function of the amino.
Yeah. Of this 20-state variable, right? Because you use 20-state variables.
Yeah.
So if it's a function of a 20-state variable,
Yeah, no, that's, yeah, sorry, go on.
is this analysis going to change significantly then, the results of it?
So I guess you're saying it's a function of xi, like it's sort of a parameter times a function of the xi's together, and it could, depending on like how like correlated the features become. So what's sort of nice about the analysis here is that we kind of assume that the features are kind of orthonormal, but if
Because,
you,
for example, there are some properties of single amino acids which are, like, for example, I don't know, hydrophobicity and so on. So basically, maybe those thetas would be also interpretable as some sort of a physical property,
Right.
physical property of amino acid sequence.
yeah. No, it's something that we're looking into, but I think the analysis would change because the features now would potentially become more dependent, or you might, and depend on what assumptions you make about how the xi's or the amino acids are drawn or generated. Yeah.
You have not shown, I think, the naive approach. You just stick it into a logistic regression model and see what happens.
Yeah. So I guess the naive approach is just that first one here. So sorry, I didn't go into the details, but that's pretty much this first one, N1, EN1. So what they do there is they just treat the unlabeled samples as negative, and then just like use that, but then just apply the fact that you know pi to sort of correct for that. And that does, yeah, so that is a reasonable approach as well. And as we can see, it does like, it doesn't do terribly, but it doesn't sort of do as well. It's basically like, it's essentially like, taking one step of the EM algorithm. Yeah, sure, yeah, thanks.
In some applications, it's a bit philosophical what labeled and unlabeled is, right? Because you can't say, if you see something, if you want it, I don't want to say label it a 0. I mean, in this particular application, it might make sense that you kind of miss out a real thing, but otherwise.
Right.
More social sciences or so. It's just encode whether you see it and the other one is just a zero, right?
Right. Yeah, so no, we tried that first and it didn't work as well, basically. So,
I mean, in this particular model, you can just make it up like this. Right?
yeah. No, that's right. Yep.
I guess I just want to follow up with Randeep's question. I think this is highly related to classification with label noise.
Right.
I
Yep.
think I saw some papers, they have very dedicated analysis on classification error rate in this case, in logistic regression.
Right.
But of course, they don't have the variable selection component.
Yeah.
So I think that might be helpful for you to derive some.
Yeah, no, I agree that there's certainly connections between like sort of unlabeled and like sort of errors or perturbations of that. But yeah, I guess no. So I think there would certainly be links there. Yeah.
Because I think in your assumption, you assume that the pi does not depend on x.
Right.
So
Yeah.
it's the sort of like homogeneous case in
Right.
some sense. Yeah, I think in this case, if you do some derivation, the base decision boundary actually does not change compared to the ideal case where you have the correct label.
Right.
So I think that actually.
No, I know. Yeah. No, and I think so. That's kind of like, I guess, gets to Peter's question of sort of you start with just like somewhere and then you just shift the decision boundary, and that's essentially what these other approaches do. But because you kind of don't really know, like you sort of have to tilt your decision boundary. That's like one step of the algorithm, and taking more sort of helps you do better. Does that sort of?
Okay, let's thank Garvesh again and all the speakers.